think the very final slide is the one that basically tells you about our different working groups. And then if you are signed up for this, it does not automatically make you a BINA member. We would like you to become a BINA member, and that QR code will take you to that, because it means you'll get Vanessa's great monthly newsletters that tell you about all the things that are going on. So you'd have heard about I2K by now. You'd have heard about all of our PD stuff coming up for 2025 as well. So please become a member. There's lots of opportunities for you to benefit from joining. And with that, I'll hand it over to Katerina, who I believe is going to be doing introduction. Oh, maybe Judith is doing this, but I'll hand it off at this point. Thanks.
Yes, thank you. So welcome everybody. I'm Judith. I'm based in Montreal, Canada. Last Monday was Canada's Truth and Reconciliation Day, so it's more than appropriate that I do my land acknowledgement statement here. So I want to respectfully recognize and acknowledge the relationship that the First Nations, Inuit and Métis people across Canada have with the land that all Canadians live on and enjoy. I recognize human rights of people who have suffered from systemic racism and discrimination in Canada. I seek to provide a welcoming, engaging, and safe community for everyone. Thank you, everybody. And now the floor to Katerina.
Hi, everybody. This is Caterina Strambio. I am at UMass Medical School. Thank you, Judith, for the land acknowledgement. I would like to introduce Kathy Wollstonecraft, who is a hero in data management. And she's actually one of the people that authored the FAIR principles. She's from the University of Leiden, and she's also leading the Netherlands Bioimaging Initiative, or co-leading. I don't know exactly her role, but she's definitely one of the most visible people in that initiative. And she's going to tell us about. So this presentation stems from a conversation we had at the AIMM meeting in Europe in June. And if you look at the wall behind Judith, you can see a big circle that represents REMBI. That means it's always front and center in our thoughts. And so we have a lot of people are working to around REMBI, recommended metadata for biological images to make sure that we can have maximum value from these recommendations. And so we're thinking that we could work together. And so Kathy is going to tell us about FAIR Don't Seek. It's as a tool that we could use for sharing some of these work that we do together around developing REMBI templates and other things like that. So please, Kathy, take it away. Thank you for joining us, and I'm looking forward to your talk.
Okay, thank you very much, Katerina, for a really nice introduction and the invitation to speak today. I'm not leading the whole NL Bioimaging initiative. I'm leading the data and analysis pillar in that initiative, but thank you for the promotion as well. No, but there's already a lot to do just within the data and analysis. I think it's one of the major components of the whole project, because actually we're starting from not really having any kind of infrastructure, and we're building on some lovely tools and resources that lots of people in the community have already provided. So Omiro in particular. But today I want to talk about the metadata management side, and we've been making use of some of the tools and ideas that we actually developed in previous projects for NL Bioimaging. The previous projects that I worked on, actually when I was in Manchester originally, so this has been going on for a while now. And we were then working with systems biologists. I still do a lot of work in systems biology, where the challenges of data integration are great, because we're dealing with lots of different kinds of omics data all the time, and we're trying to link that with models. So this is where the FAIRDM-Seek project originated, and I want to start by giving you a bit of the background of that and how we came to design it, and then how we now use it in NL bioimaging and where I think it might be useful for all of us in the future. So the FAIRDM-Seek project, we started in 2008, so it was really a long time ago. The FAIRDM part stands for FAIR, Data Operations and Models, and it's a community-driven research infrastructure, so we were developing it for very large systems biology consortia, quite a few different systems biology consortia, and you can see some of their logos on the screen. But these were large projects where lots of different people were working together in labs spread across Europe, usually. And we had multiple different projects within each consortia. So we had, I think, 113 different labs in the first consortia that we worked with, and that just got bigger and bigger over time. There's a big team that developed this in Manchester, in Heidelberg, in Stellenbosch, in South Africa, Zurich. These days also in Belgium. There's lots and lots of people working on this. There's some more logos there. And the leader of the whole project is Carol Goebel from Manchester, and she's also the head of Elixir UK. And because of that, the FAIRDM-Seek platform has also become part of the Research Data Management Toolkit, which Carol and others are leading. But how did it all start in the first place? Well, if you go back to 2008, as a systems biologist, you were generating lots of different kinds of omics data. If you wanted to do good research practice, then you had to then submit different parts of your experiment to different repositories. So there were a lot of public repositories available. Each one had their own minimum information model specification. But the only place where the experiment as a whole came together was in the scientific publication, which wasn't very computationally amenable. And if you looked at any one of the outputs of the experiment, the rest was not visible. And so systems biologists, they really wanted somewhere to actually share their work and their experiments. There was a lot of data that was being generated where there was no home for it as well. And so there was no repository, there was nowhere to put the data. And this is where we started with the FAIRDM-Seek. And we just wanted to address that central problem. This was way before the FAIR principles came into being, of course. This was just about packaging and gathering together the different information that was being generated in these experiments. And if you think about the way that systems biology works, you have experimental scientists on one side and modelers on the other. Sometimes people do both, of course. And you have informaticians working in between to do various kinds of analyses. And this process of doing the research is quite cyclical. So all stages through the research, you need to be able to share the data, the models, the protocols, every kind of research asset you can think of. But you don't want to share with the whole world straight away because it's not been published yet. So we needed a flexible environment that would allow pre-publication sharing. And then once you had published your data, then post-publication submission to various different public repositories in a straightforward fashion and more general dissemination and sharing. So that's what we were aiming for. And that's what we generated. So the FAIRDM-Seek and the FAIRDM Hub, which is the public version of that, which is hosted in Heidelberg and is available for anybody to use, actually. Predominantly, we have European researchers in there, but actually it's open to all. And it's a research assets catalog. So you can use it as a repository, you can upload things there, but you can also point to resources that are in other places. So that could be the public repositories, it could be local resources that you don't actually share, they're just pointers for you, so you can keep this context of your whole experiment. They could be pointers to restricted stores that only some of your project have access to. And all of these different combinations are also possible. as an individual scientist, you are in control of who sees any of your data at any time. So you can share it publicly, or you can share it with named individuals, named projects, and various other granularities in between. Everything that you upload or share can be attached, it can have a license attached. And sometimes you can say, I only want to share it after this date, or I only want to share this for a certain limited time so that somebody can review this data as part of a publication review process, and then the link expires. So it's very flexible from an accessibility point of view. But the core of what I want to talk to you today about is interoperability. So how do we achieve this interlinking and helping scientists keep the context of their experiment, and help scientists share their data in a much more understandable way for the rest of the community? Well, at the heart of the infrastructure is something called the JERM, the Just Enough Results Model Ontology, and it's an application ontology. So it's a very simple, straightforward, pragmatic approach to describing all of the different research assets that we deal with in systems biology, but also in science more generally. And then for each data set that you upload, or model, or protocol, you can say what type it is. This example here is about data. So if you upload some RNA-seq data, for example, you can say it's transcriptomics. You can say you're measuring gene expression. You can say it's time series or it's not time series. And you can describe all of these properties. Some of them actually come from the minimal information model anyway. You don't have to do double work. But the idea of the GERM is that it pulls out all of these common entities that exist in all of these different minimal information models. But then that's still just working within one minimum information model. We also use the ISA standard, Investigation Studies and Assays, to help us link in between. And ISA is a very simple structure to understand as well. It just simply says these particular assays belong together in this study. In this study, we're using the same biological material. And this is the relationship between all of these different experiments. For us in systems biology, we extended this a little bit to incorporate the idea of modeling. So you can also say this data was used to construct a mathematical model. This data was used to validate this model. These data sets should be completely separate if you're doing good scientific practice. And this is now a little bit more generally applicable, actually, because there's all sorts of different types of modeling in all sorts of different types of science, and especially in the AI world. So you also need to describe your AI models in a similar sort of way. But through observation with our end user scientists, everybody used Excel spreadsheets or just simple tables to store and share their data locally. And instead of trying to persuade them that they should be using some kind of complicated database data entry system, we just simply went along with that process. And so actually what we did using this GERM ontology model and the existing standards, we developed a series of spreadsheet templates to describe different kinds of data, different kinds of modeling, and we propagated these templates. So we shared them within our research communities. We shared them actually using the FAIRDom hub to share them more broadly. And they are starting points for a particular community to take and modify and extend in the way that they need to. So we have a Just Enough Results Model master template for different domains, and then people extend these. Now, to make this a little bit easier for people, we also thought about how you might embed additional semantics. And we developed a tool called Writefield for this. And Writefield simply takes one of these templates that we've developed, and in places where a minimum information model specifies a particular metadata, well, no, not metadata, but a particular ontology to use for your annotation vocabulary, then you can embed collections of terms from that ontology into a particular cell in the spreadsheet. Now, this is an administration job. It's not an end user job. The end user sees an Excel spreadsheet with some drop-down lists, and all they have to do is select the right term from the drop-down. And then when this spreadsheet is uploaded back into the FairdumSeek, the metadata is extracted into RDF in more of an automated way. And depending on how you set up your FairdumSeek instance, determines whether that's directly searchable or whether that's searchable through some kind of interface, or whether that's just hidden away from regular users. It's entirely up to you. But by doing things this way, we can introduce more standardization and richer semantics without users having to change exactly that much about what they're actually doing day to day in their research process. And this has proved quite popular with our research communities. The Right Field tool is in urgent need now, I think, of a version 2. We've reached some kind of limits of scalability in terms of the sizes of ontologies. And actually, we just want to do more things with it now. So we want to say things about how different groups of cells link to other groups of cells within the templates. And so I would dearly love to develop a version 2. We've been starting to work on that, but it's a very slow process. It's still a usable tool as it is, but I think we could do an awful lot more with this approach and with this tooling. Now we have this tooling. This is the ultimate, the consequence of having something like this. It doesn't matter what kind of data you start with. It doesn't matter what kind of minimal information model that data requires, where the data, the metadata elements actually overlap. You can aggregate. Where they don't, then they don't. But you don't have to redesign your data model every time you add a new type of data. And this is very good news, because actually people are developing new types of data all the time. People are developing new kinds of omics approaches. And so we need this flexibility to actually future-proof the whole system. And that's why we went for this semantic approach in the first place. I've been talking a lot about data, but of course, this kind of semantic annotation applies to models as well. So this is an example of one of our models displayed in a tool called JWS Online, but you can see actually that we're using the same annotation vocabularies in the models. And that means on top of that, that we have all of this standardized data that we can more easily interlink behind the scenes. There are lots of tools that plug into the Fairdum Hub. Quite a lot of them are about systems biology modeling at the moment, but that's expanding and extending. There's a link to Galaxy Workflows now, for example, so you can do all sorts of informatics processing. And the results of these processes can be interlinked into your ISA structure as part of your experiment as well. And if you think about this from a publishing point of view, when you get to the point where you do want to publish your data, then you want to be able to put together various groups of files, data, models, and other things, and package them in a way that is understandable and amenable to future use. And so this is what we have. You can create a research object from collections in your Fairdum Hub directory. And this is all based on the ISA standard, but once you've collected together all of the things you want to represent in one particular publication, you can give that collection a digital object identifier. So then it's fixed, but it doesn't mean that you can't continue to develop those models and those data sets later on. So this paper actually comes from quite a few years ago. You can follow the links and you can find the DOI and all of the data and the models that came out at the time. But this group continued to do this research. And this is the latest publication from this group. You can see how it links to the older models and data and how those models have evolved over time. And you can see the relationships between all of these things. You can also see a new DOI for this particular collection of research. And actually, if you click on the actual figures in the paper, you're taken to live versions of the data and the models. So you can actually do your own reanalysis if you want to. And so we don't want to create a kind of great data graveyard. We want to create something that's a living, breathing representation of what people are doing in their labs and help people share and propagate good methods and interesting results to the community. Well, this seems to be working. We can track how many times people use various research assets in the public FAIRDom Hub. So we just look at how many views and downloads we have. And here are some examples. As a PI, this is very useful because you know where all of your data is at any given time, which is something that you are required to do most of the time now. And so pre and post publication data is organized and available and accessible to you. So if we think about this from a more general point of view, I described this cyclic experimentation process between the experimentalists and the modelers and the bioinformaticians. And this is not too dissimilar, I think, to the type of thing we see in bioimaging. We generate images, but actually then there's a lot of things to do to those images before you get the biological insights out of those images. And that quite often involves multiple different types of researchers working together. So here I don't think it's really dissimilar at all. This picture comes actually from a paper in 2016, but the process is still the same now. And both of these processes are also not too dissimilar from just the regular research data management life cycle that probably we all know here anyway. And Katerina mentioned the REMBI metadata standard at the beginning. The REMBI also uses this research data management life cycle, and it specifies, again, a minimum information model that we can follow in a similar way to all of the omics ones that I already described. So for us, this seemed like the same process that we needed to go through in NL bioimaging to help us understand how to share our experiment metadata in a better way. And so this is what we've been doing. We've been using the same mechanism of developing templates with embedded semantics to develop REMBI compliant templates for various different domains of microscopy. If you look at the REMBI specification, which I'm sure many of you have done many times, it's quite general, and it needed to be quite general because it had to support the whole community. And there are various working groups to make it more specified, but we need to go even further than that and make practical, pragmatic, concrete instantiations of some of these different working group outputs to actually move people forward in the community. And this is what we've been doing with high content screening. And where you see here in the REMBI specification, there are various different ontologies you can use to describe certain parts of the specification. We've just gone one step further and said, well, we've chosen one. This is the one we've chosen. And we've chosen, these are the terms you need at this point in your data annotation. And so, well, here's an example. So again, we have these drop-down lists where you specify what a user can say at any given point for that particular type of microscopy experiment. And we've done some work on mapping the REMBI specification with ISA. We did this specifically for the high content screening domain, but actually this is higher level, is basically REMBI mapped to ISA. So we can now make use of that for other bioimaging domains. And that's what we're busy with at the moment. I think some of you have already seen the paper that we produced about this work. If you're interested, we have all of the examples that we've developed and presented using this new specification in the FAIRDom Hub. So we're using it now, not just as a data store, but actually as a way of propagating the templates. So the templates are there and some examples of how we've made use of those templates. And we're going to continue to use this for distributing templates from other related domains as well. So this is where we are up to now. We have the FAIRDom Hub to propagate the templates that we've developed. These templates can be versioned. You can track who is using them. You can see how they link to publications. So we can keep track of how well this is going in the community, if you like. We can try and use this to promote community consensus and share common representations. And we can start to try and use this to facilitate brokering and migration as well. So at some point in the future, it would be really great if we could press a button and submit to Bioimage Archive. Of course, this is mostly just metadata, so there's some data somewhere in an Omiro server somewhere in Leiden as well, and the two things need to be packaged together and sent to the Bioimage Archive. And this is something else that needs some work over the coming weeks and months. So with the FAIRDom Hub, there's a lot of work going on at the moment on developing RO-Crate specifications and RO-Crate extensions to the platform. And RO-Crate is just a way of, well, it's a lightweight solution to packaging together metadata so you can describe your data and where it is in a more portable way. And we in Leiden are also thinking about this from the point of view of what that means for bioimaging data. So there's the work on actual RO-Crate support at the platform level, but there's also the work on what would an RO-Crate for bioimaging actually look like, and we've started to think about that now. We've started to think about all of the things, the extra things we need for extending this idea to other kinds of bioimaging. So we are looking at developing a more formal semantic model for the model that we have with REMBI and ISA now. We've been looking at extending the germ for bioimaging terms. I think there's some work to do more generally on ontologies in bioimaging. We're also thinking along with new communities of bioimaging researchers within the Netherlands to think about how we accelerate this generation of templates for different kinds of images. So I think probably that's where I should stop talking. So we have a bit of time for a discussion, but I hope that's given you a good idea of what we're doing with the FAIRDom Hub, why we are using it for bioimaging, and yeah, maybe some of you would like to get more involved with that and join in with community efforts.
Thank you so much. This was everything I was hoping and more. So thank you. So there is so many things to talk about. So I will open the floor to questions and comments. And so Josh has a question. Can templates be extended, and are templates different from models?
Yes, so yes, we really would like for people to extend templates. We would really then like for people to say how those templates relate to the templates that they got originally, and therefore what's the provenance of that extension. When I'm talking about models, I'm talking about the mathematical models, so the representation of things like glycolysis for how various different concentrations of enzymes and small molecules are propagated through the system as you perturb it. So it's a different, the templates are the metadata, and the models are a different kind of data.
I think Josh was talking about metadata model. So
I see.
I do have that question too. What is the relation between, so what do you think is the relation between a template and a model? I guess based on what you have said, my paraphrasing would be, the template is an instantiation of a model, a metadata model. But I would like to have what you would say.
Yeah, so I think there's quite a variety of formality and specificity across the whole of the life sciences. When people talk about the minimum information models, actually sometimes they're just really quite loose checklists, and sometimes they're very well specified metadata models where they say this is the format, this is the annotation vocabulary, and all things in between. So it's the templates for us are an instantiation of our model. Sometimes we're formalizing it, sometimes we're just following the specification.
Sorry, I actually, Josh, could you just, we cannot assume that everybody knows everybody. So Josh, could you just in 1 minute say who you are? And then I'll ask Alex who asked the question also to say who they are before they ask their question. Sorry, Josh, I just missed that before.
No worries. Hi,
we're gonna do it in reverse.
And I guess in this context, so I'm here with an OME hat. I guess I could change my background. Excuse me. So a core OME developer, so responsible for developing OME XML and IDR and Omero and all that. Hi, everyone.
Hi.
Hi, Josh.
And Alex, if you want to introduce yourself and then ask your question, that would be great.
Sure, I'll be brief. So I'm with Thermo Fisher Scientific. I have a background in museum science and also have done sequencing work, so I'm familiar with some of the data repositories that are used for sequence data. And I also have an interest in long-term storage of image data. So this scratches a lot of itch, you could say. So this is why I have the two questions in the chat, which are related to the long-term curation and storage of all of this information. It looks very nice right now. It's very functional right now, as far as I can tell. Who is responsible ultimately for the long-term storage and maintenance of the data? And then a related question, are there any risks to that long-term storage as far as you see it?
Yeah, so both very good questions. I think that what we always try to promote is that data shouldn't be stored using the repository function after publication in the FAIRDom hub. It should always go to the public repository that it's supposed to go to, and we maintain the links and the context. So the actual data should be somewhere else ultimately, but the actual platform is guaranteed for at least the next 10 years by HITS, the Heidelberg Institute of Theoretical Studies, which is a non-profit organization in Germany. So yeah, sustainability after that, of course, will at some point be a problem, but actually we've been guaranteeing this for 10 years for at least the last 10 years now. So so far so good. That's the best we can do, I'm afraid.
Yeah. So this issue of storage and risks and all of that, it's a big topic. And in the US, in North America, we're just starting to deal with it. I was just having a conversation this morning with people from IT. Often we have we still have to deal with coming up with data governance policies, and which is and everybody is like hoping that somebody else will think about it and will guarantee it. So I mean, this is so seeing that there are people like you and other success stories, it's a good I mean, even though, I mean, you haven't solved every single thing, because you say, I can guarantee for 10 years, but 10 years, it's an enormous length of time for somebody that has nothing at the moment. So that's, it's a very good success story to think about and to look to get inspired by. And okay, so Judy.
Well, I just wanted to comment briefly in reaction to Alex's question. In Canada, the government did implement a whole infrastructure for data management. And we're not there for microscopy, but I can tell you that for astronomy data set, it's been supported for several decades. So we're working actively to hope to get to the same point with microscopy. Thank you.
Yeah. I have a question. Does anybody else want to chime in? Otherwise, I'll ask a question, and maybe that will spark more comments. So, so one of the reasons why, I mean, I'm very inspired by your work on data, but specifically right now, I'm interested in the work on metadata and the templates, the right field, using right field as a way of enriching those templates with ontology. And that it's all stuff that I think we should use as a model to bring, to reach the users. Because at the end, we need to bridge between models, specification, recommendation, guidelines to a concrete thing that users can have when they're at the microscope to enter information once and not ever again, hopefully. So that yeah, and so it's really an important aspect of what we're trying to do. And so the idea of using Fairdom Hub as a way of sharing these templates is it's something that I would like to ask you. I mean, you're of course you're doing it, so obviously I would imagine you would recommend that could be like a place where other people could do it, and we can have a more general sharing of templates. One thing that specifically, though, I wanted to ask you is one idea with REMBI is that there is this different metadata for each step of the data lifecycle, and we have been calling each of these steps, we've been calling it REMBI module. And so one could actually go into the granularity of saying, let's share REMBI modules, not just the full template, so that editors, like the people behind the scenes, could more easily create a template for individual users by, for example, taking a study component, and then maybe they have to tweak the sample and the specimen, but they don't have to start from scratch. So how would you think that it would be possible to do that with Fairdom Hub, so that we can actually have a granularity of sharing, REMBI study templates and REMBI study component templates? How would you do it? And do you think that would be an appropriate place to do it?
Yeah, I think it's a good idea to reduce the granularity a little bit like this, so that you make these templates more modular. We've been talking about the metadata like it's complete at the moment, but actually there's a lot more work to do, I think, on the standard when it comes to the analysis part anyway. So these are not really complete templates already. So I think there are a number of different ways we could tackle that using the FERDM Hub. I don't know at this moment which would be the best way, because we're sort of borrowing the ISA structure that's already there. So I suppose the components would be at the same level as the assays are in the current structure. Yeah, we would have to see if that was still appropriate, or if we would have to think again about where they should go. But I would say that's where we should start. So you could have sets of components represented as assays for a particular kind of bioimaging technology. Yeah,
because right now FERDM Hub has in it the structure of ISA, so investigation, study, assay.
Yes.
So if you want to say, for example, different modules, okay, if you want to have different study, well, I guess because a study could be at the maybe in the investigation. So so we would have to think about it. Okay.
Yeah.
And have and maybe we can follow up this meeting with another more in detail to start. We can actually talk about how we would implement this, because I think there is a lot of people that are already that are working on this template thing. And by the way, I want to also emphasize that, yes, I mean, you gave a very good explanation why you're using Excel spreadsheets, and I totally agree with that explanation, and so that's what we have started doing. But I should also say that at the end of the day, these templates are just collections of key value pairs, so that they can also be implemented in other ways. So that's the other thing that I want to be very clear, that if, let's say, we create a RMB study set of key value pairs, those can also be implemented in a database or in any other way. People are like very much Airtable these days. I mean, anything that allows you to implement it doesn't have to be an Excel spreadsheet, as long as we all start converging and maybe try to build consensus around examples, and then other people can use the same sets of descriptors but implementing in a different way. I think the idea of using Excel as a starting point, it's a good idea, because anybody can download it, anybody can start using it. While an individual institution can have other solutions which are more complex and more usable, better for users. But anyway, so so I just want to be sure that people don't think that the idea is that we are trying to sell Excel to everybody. It's just the idea of standardizing around terminology and templates that could be used in different contexts. Okay.
Yeah, I completely agree. And we try and represent the data not as key value pairs, but as triples, so that we can use RDF and the semantic web technologies for integrated search as well. But yeah, Airflow is becoming very popular. I think that would be a nice thing to explore as well.
But just to follow up on so triples, but that's behind the scenes. As far as the user, it's they only see the key, the field name, right, and they enter something. So as far as they're concerned, you deal with it with a triple, but they don't see it. So that's a good thing.
Yeah.
And yeah. So any other comments?
So Kathy, you said that you were working again on right field. Is that correct, or that's future plan?
Yeah, it's an ongoing plan that's stalled at the moment because of personnel moving to a new job. Yeah, watch this space. Hopefully we will get some new resources for starting that up again.
Excellent. I look forward to it.
Yeah.
And maybe, you know, same issues. Everybody has issues with limited resources, but I mean, hopefully, if we can build more resources, we can also maybe we will be able to be in the position of contributing. So anyway, just to explain what right field is, if people, it's basically like, it's a way of embedding ontologies in an Excel spreadsheet. So again, the user doesn't need to do much. They just need to select fields, values for a certain field that come from an ontology. And then behind the scene, the software knows that there is that term corresponds to a specific ontology term. So it is very powerful, but at the same time, very easy, make it easier for the user. Okay, Josh has another question. Can you please ask it?
Okay, I mean, if we're talking about resources, what would you want from the bioimaging specific software to work better with Seek? Like what would, you know, the perfect future, like where do we go?
Yeah, so well, I think from our setup where we have 18 different institutes and everybody is generating their own data and storing it locally, we need some federation. And so I think we talked about this before, ways of packaging things together and pointing to them or exporting them to some other platform somewhere else. I think that would be really useful. So this, I think we talked last time I saw you about arrow crates a little bit. Maybe this arrow crate specification would help us get to that point. Yeah, but I think you being able to say, actually, this is my series of experiments and some of them are local, some of them are already published. But some people should be able to access all of them. That bit is the tricky part. And I think there are mechanisms within the Netherlands for sharing credentials across institutes. And we're exploring that at the moment. So some of that problem just means people will log in to somebody else's Omiro and use it that way. But there's, yeah, I think being able to package and send collections of experiments to other instances of Omiro, to other places would be really good. And I think, well, the new file formats, OMI, again, should help deal with that.
Yeah. Just yeah, along the line of this, Josh's question reminded me of another question is, how do you currently, so apart from, you know, things that you would like to see, but how do you currently make sure that this, let's say that you are doing like a high throughput screening, you have now this recommendation, minimal recommendation for it. So then there are the templates. So how do they end up annotating the images in Omiro? Is that link currently done? Do you attach the filled-in template to Omiro, or you import the key value pairs? What do you do? What do you guys are doing now?
Well, we're actually exploring both options. We're attaching those templates to Omiro and also the electronic lab notebooks that we have in Leiden that everybody is supposed to use. So then people don't have to fill these things in twice. It's just the once, and then we link to them. But yeah, there is some work going on. Somebody else in the network is doing this to how you might then go about converting from the template to the key value pairs in Omiro directly. Yeah, the tricky part is actually the social engineering. How do you make sure that people do this at some point during the experimental design? In Leiden, we have a system now where when you book a microscope, somebody will say, have you registered your metadata? And this is when they are required to do some thinking about their metadata and data structures before they do any imaging. But it's not completely enforced, so people can find a way around it if they wish to. I'm in two minds about whether we should really enforce it or whether that would be just too much for scientists, and maybe gentle encouragement is better most of the time.
Yeah, so we are working to one of the reasons why we have started thinking about templates is because we are developing a tool to import to transfer the key value pairs to Homero and what we are experimenting with in Canada and in UMass is the fact that you do that automatically when people acquire the images. There is an importer that scrapes the data and the metadata and brings it in. So they enter it in the. I mean, the thing is that there is really no direct link to the electronic lab notebook, that it's available at the sites that we are dealing with. And so the idea is we are experimenting with the idea that, you know, if we give them, if they're going to use an Excel spreadsheet anyway, if we give them, and often this Excel spreadsheet, each person in the lab has a different one. If we give them a common one for the lab and we work through it with them, and then they will say, okay, I like this, and they'll start using it because it's something they have to do anyway. And so we are seeing some success with that idea. Like, you know, I might as well use this template because it's more organized and it's at least everybody else in the lab will use the same. And then it moves out from there. So, and people do like it because I can start entering the information as I'm planning the experiment. I will have to do it anyway. And I might as well do it in a format that then I can find it with the images afterwards. So, but it's, so enforcing without having, I mean, in your case, you have something already working, so that could be, you know, maybe down the line, there's going to be some enforcement, but for now, we just have to do, added, you know, making sure that people see a value to what we're asking them to do immediately, rather than thinking about sharing in the future. It's just something for them right now. A gift to themselves, to their future self. I think Caroline came up with that idea. Gift to your future self. Metadata is a gift. Metadata is a gift to your future self, and I think that is so true.
Yeah.
Any other questions or comments? We're getting close to the end of our time. If not, I really thank you again so much. This was amazing. And we stay tuned. So we are so this meeting was you know, basically shifted down 1 month. So now we will have to decide whether we're gonna do it in December, and the next one sorry, Vanessa, go ahead.
Yes, Katerina, if I may, we already have a scheduled presenter from the Allen Institute.
In November.
In November. So
okay,
yes.
so the next meeting is November then. Sorry,
yes, November 1st.
Okay, perfect.
Yes.
So we already are all set, and I was just forgetting. Sorry about that, to generate confusion. Okay.
Natalie, do you want to share really quick?
Yeah, I'd just like to say something. So Sean will be presenting from our institute. We have developed a tool that fits so well with everything we've heard today. Actually, thinking we should probably collaborate a little bit. You're missing a piece and we have it, basically. So I think it will be very interesting for everyone.
Great. Fantastic. Okay, so we're looking forward to that, and see you in November.
Excellent. Thank you so much,
Bye.
Kathy, everybody. We're getting there.
Yeah, thanks for having me.